So welcome everybody. Thank you for being here. It's really a great pleasure and an honor for me to welcome Yannis Ioannidis at our institute. As you may know, Yannis Ioannidis is an honorary member of IMBB. He was elected earlier this year by the board of directors of Ford. And he is coming to give us a presentation today in that capacity. He will be properly introduced and presented by Fotsis, who came deliberately from IOANNIDIS for this event. So I won't really take much of your time. Just to say, we're happy to have you here. We hope to see you again soon and frequently. And just as a small token of our appreciation, I would offer this to you on behalf of the Institute of Molecular Biology and Biotechnology and the Foundation for Science. So,
a small introduction. John Ioannidis was born in New York in 1965, so he's not even still 50 years old. He grew up in Athens and he graduated from the Athens College, first in his class. He studied medicine in the Medical School of Athens, where he graduated again first in his class. In 1984, if I'm correct, he won the first prize in the Greek Mathematical Association, or the Association of Mathematics. He got his PhD degree in 1996 from the University of Athens, and in the decade between 1990 and about 1999, he specialized in USA, in Harvard and Tufts, in internal medicine, infectious diseases, and clinical epidemiology. And he also served in NIH in Baltimore before returning back to Greece into the University of IOANNIDIS as associate professor. So in 1999 he returned to Greece as associate professor, and in 2003 he became professor of hygiene and epidemiology in the Medical School of University of IOANNIDIS. All during this time he was director of the lab. At the same time, or a little bit later, he became also a cooperating member of the Institute of then called IMBB, which was the name of our institute before merging to IMBB. But unfortunately, in 2010, he started moving away from Greece and back to the States, and he went to Stanford, where, as you see, he currently holds several positions in the University of Stanford as professor in prevention of health disease prevention, internal medicine, research, and I have it here in Greece, and policy, research and policy and statistics. Apparently statistics, it is in the faculty of, not in the medical school, but in another faculty. At the same time, he is director of two institutes, the Institutes of Research in Prevention, and he co-founded with others a center of meta-analysis, so-called metrics. I will not go into details into all associations and councils in which he is member, because we will not finish today. He is in the editorial board of many, or at least 30 journals, and he is chief editor of the European Journal of Clinical Investigation. He has given more than 300 invited talks, but what is very important and very interesting is that many people think that all his work was done in the States, and that's why he was called back to the States, because of the work he was done there. But his work was done actually in IOANNIDIS. When he came to IOANNIDIS, I was in his election as a lector. He had about 3,000 citations, and when he left, he had at least 10 times more, 30,000 at least. So all his work was done in IOANNIDIS, and based on this work, he was called back to the States and to Stanford. So it's not that he did his work in the States and he came back. He did his work in Greece and he was called to the States. And a considerable part of this work was done in our institute, where at least 10,000 citations have the name of our institute. And therefore, he is now an honorary member of IMBB and ITER. So today, he will speak about improving reproducible research, and I'm delighted to call him to do this talk.
Thank you, Theo. Thank you, Nakaya. Thank you for the very kind invitation. It's a great pleasure to be part of the fourth family. I feel extremely privileged for this honor today. And I have to say that it's not news that fourth is the leading force of science in Greece, and one of the leading forces of science in Europe and in the world. I think that everybody in this auditorium should be very proud about all the work that is being done here. A few months ago, actually, I published a book in Kedros editions, where one of the chapters has identified systematically all the highly cited, top-cited Greek scientists around the world. And this is how it looks like. 336 people were in the top 0.5 in 1,000 of the scientific community in terms of citation profile. And if you look within Greece, the highest concentration, the highest number is people affiliated with fourth. If you use a denominator of a population basis, even though we have a few more in California, if you divide by the population, there's no place on earth that extremely influential Greek scientists have concentrated. And I think that it's not just Greek scientists. I think that fourth is famous also for being able to attract talent from very different countries, just because priority is given to science and high level of competitive research. I will try to give you some views, some of which are summarized in an early paper that I wrote from IOANNIDIS about 10 years ago in PLOS Medicine, that since then has been downloaded about 1.2 million times and has been cited several thousand times. And also a more recent paper that is trying to propose some solutions about how to improve the reproducibility of research. So why are we not able to deliver the best science that we want to deliver? And how could we improve practices in science so as to make more published research true? If you take a bird's eye view of science, there's more than 15 million different scientists publishing papers in major scientific journals, just based on Scopus in 1996 through 2011. Within that time, they published about 26 million papers. And if you extend the time period and databases, there's about 114 million scholarly documents across all science and all times. Each empirical paper can include anywhere from a few up to many millions of results. So we're talking about billions and trillions of scientific results. And there's also even more that maybe were not considered good enough or high priority enough to be written up. We're talking about a treasure of information. How much of that work really makes a difference? And how much of that work is trustworthy so that other scientists could use to build their own research? How could we decide? What should we look for towards ensuring that more published research is credible? Should we look for significance? Should we look for innovation? Should we look for rigorous, that means reproducible, approach to research? Most funding agencies are asking for significance. So if I have to submit an NIH grant, there's one major item that says significance. You have to convince the reviewers that your work is significant. In Europe, it's pretty much the same. But almost all scientific papers claim that what they have found, statistically and or conceptually, is significant results. And obviously, all grant proposals claim that what they do is highly significant. Recently, along with David Chevalarias from the École Polytechnique in Paris and the Institute for Complex Systems, we looked at PubMed. We found about 1.5 million abstracts with p-values, and 96% of them reported statistically significant results. So, if we're asking for significance, everything is significant. It has lost its discriminating ability to tell us what is important, what is true, what is useful, what is translated. How about innovation? We also seek innovation, so one of the other boxes and one of the other criteria in all NIH funding, and obviously also in Europe, in ERC grants, we want to be innovative, disruptively innovative, ideally. But is there any method to predict innovation ahead of time? It's very difficult. The vast majority of honest and excellent scientists never produce anything that is highly innovative, despite continuous hard work and every good intention. And innovation takes many years, often decades, to materialize and to translate itself. A few weeks ago, along with some colleagues, we published an analysis in Nature where we asked the most highly cited scientists across biomedicine around the world to characterize their papers in 6 dimensions: continuous progress, broader interest, greater synthesis, disruptive innovativeness, surprise, and publication difficulty. What they said is that most of their most highly cited work was so important not because it was disruptive, not because it was a big surprise. It was incremental progress, broader synthesis, continuous progress, and not that much of a revolution necessarily. But we also performed an analysis where we looked at these indices in terms of their correlation pattern. And if you see a red link, that means that there's a positive correlation. If you see a blue link, it means that there's a negative correlation. There's no blue links here, which means that these dimensions of revolution, of disruption, and surprise, and unusual, unexpected, versus continuous progress, greater synthesis, seeing the big picture, are not contradicting each other. They complement each other. How long does it take? This is another analysis that we had published from IOANNIDIS in Science about 6 years ago. We took the most highly cited papers in clinical medicine, those that led to major applications for medical practice, and that had been cited more than 1,000 times. And we asked the question, how long had it taken in the past to get to the point of having that landmark paper that was so heavily cited? If we take a look at, I'm not sure whether there's a pointer that I could use. we can find one. But if you look at the red lines, these are the highly cited papers. If you see some black lines in the top part, these are papers that actually were refuted down the road. So, and we thought that we had found something major, and that was not really the case. So many of those actually were refuted, but regardless, it took many, many years from the time of the first description of the intervention, the basic discovery, until reaching the point of the clinical translation. On average, it took about 25 years. And in some cases, like nitric oxide, it took about 200 years. So if we try to decide what is important research for credible research or reproducible research based on its innovation and translation impact, we really have to wait for a very long time. It's very difficult to judge it immediately as you see an application or a paper. This is another empirical evaluation where we looked at papers published 25 years ago in Science, Nature, Cell, Journal of Experimental Medicine, Journal of Clinical Investigation, Journal of Biological Chemistry, that clearly made a promise within the paper that what they had found, what they had discovered, would have a major impact on clinical medicine or public health. And try to see how many of these promises materialized and how long it took. So this is the proportion of these claims that had a randomized clinical trial or a positive randomized clinical trial. So a few of them did have that down the road, but it took a while. It took 10, 12, 15 years, and after 10, 12, 15 years, there was a plateau effect. If they hadn't reached that point, it wouldn't really happen later. Among 101 highly promising novel technologies that we found, only 1 in 4 resulted in a published randomized trial. Only 1 in 5 was validated in at least one positive randomized trial. Fewer than 1 in 10 entered routine clinical use within 20 years of the basic science promise. Only 1 has been used extensively for the license indication. And is that bad news? Should we despair that the translation rate, the effective translation rate is 1 in 100? I would say this is what happens. It doesn't mean that the work of other scientists that didn't result into a translatable discovery should be dismissed. We should celebrate the scientific effort in total as a community, and some may be a little bit more lucky down the road compared to others, but everybody has contributed. The main issue is, can we make that 1% become 2%, or 3%, or 5%? Maybe by improving some of the connectivity of the research efforts and the efficiency of some of the steps that we do take every day in conducting research. Let's take a look at what is the typical recipe for a researcher in most scientific fields. Typically, we're talking about a principal investigator, solo, siloed. By default, since he or she is alone, he or she can only run small size studies, struggling to get significant results, so cherry picking one or best hypotheses, mostly post hoc, searching of the space of data. A p-value of 0.05 is enough. No registration of the hypotheses or protocols. No replication. If you say that I'm going to replicate what others did, most funding agencies will say, why should we fund that? Someone else did it. No data sharing, because data are seen as a gold mine that you have to use for your own promotion, publication, advancement. And if you share it with your competitor, then he's going to be funded and you're not going to be funded. So this is the typical recipe. And I'm not saying that it's happening in all fields, but many fields have operated with that prototype. And empirical fields, and empirical studies on fields that have operated with that recipe have shown that the efficiency is very, very low. How do I know that? Well, we have some examples from fields that transformed themselves and said, no, we want to fund replication. We want to check what happened when you had all these single investigators proposing one hypothesis at a time and one publication at a time. We want to run very large studies, 100 times larger, and see what happens. Who's going to do that? Who's going to fund replication studies that are 100 times larger? Well, in genomics, the price went down so sharply. The price of running genotyping decreased about 100 million-fold over 20 years, that we could run very easily replication studies of the entire literature of candidate gene associations that had been proposed in tens of thousands of papers in the pre-genome-wide association study era. When we did that, we saw that most of the initially claimed statistically significant effects were false positives or substantially exaggerated. In the case of human genomics, now that we have gone through the revolution of genome-wide association studies and also whole genome sequencing studies, going back to test what we thought we had found in the candidate gene errors, we know that the replication rate is 1.2%. So 98.8% of what we thought we had discovered with the paradigm of the siloed investigator were false. Who else might want to fund replication studies? A pleasant surprise, the industry. I think that I have been a very fierce critic of the industry over the years, but in the last few years I have found some wonderful colleagues and allies in the industry who want to promote better research, more transparent research, less biased research. Well, it sounds very weird, but why would they want that? Well, they want it because they want to promote reproducible research for their research and development. If they use academic research that does not translate, and they cannot repeat the experiments of academics, then they get nowhere. So Amgen and Bayer and other companies have tried to replicate the best studies by academic investigators, and their replication rate is somewhere between 10 and 25%. The failure to win the war on cancer has been blamed on many factors, but recently a new culprit has emerged. Too many basic scientific discoveries are wrong. Which country might be worse? What do you think? It has become part of a monologue so far. Would you think that the reproducibility problem would be worse in the US, United States, or in Europe? Any thoughts?
More competitive, more US.
Yeah, any contradiction to that he's proposing? So indeed, in that paper in PNAS, we found that exaggerated effects were more common in the US. And the US has been more competitive in terms of asking investigators to promise more and to deliver extremes so as to be able to say that we found something. Now obviously this is changing. I think that competition is happening all over the world. That paper came out last year, but the data have been collected over a couple of decades. Another parameter that is changing the landscape is the fact that we have bigger and bigger data. But at the same time, that could also mean big noise and also big error. And last week, along with Wynn Curry, we published that perspective paper in Science, where we tried to navigate through some of the promises and some of the problems that we face now that we have an exponentiated level of information compared to what we had in the past. How could we solve some of these problems? There's many solutions about these challenges that I mentioned. Some of those have already worked in specific fields and may need to be considered in other fields as well. Other solutions are more speculative. I have to say that empirical evidence for their efficacy and efficiency is not always there. I think that we need more of that. And we have to be careful because seemingly effective solutions may also have collateral damages. So If I say, double research funding, wonderful, I fully agree. Could we have collateral damages from suddenly doubling research funding? Or if you say, cut research funding in half, I mean, you can easily see huge collateral damages by that, and try to enforce quality, better quality. Each solution has to be very carefully thought and make sure that we do no harm. So 2 months ago in PLOS Medicine, I published a paper where I tried to go through some of the main types of solutions that have been proposed, and I will run through a number of those. Large-scale collaboration and adoption of replication culture. In genomics, there's no distinction between discovery and replication. Discovery is replication. Replication is discovery. It all happens in one step. It happens in big teams of scientists who are all both discoverers and replicators. And this is genomics of smoking cessation and smoking behavior. Before we did that, we thought that we had found 400 genes that regulate whether you smoke or not. When we did that, we realized that all of those were wrong. And now we have 7 genes that we know do regulate smoking behavior with very, very rigorous statistical evidence and very good biological rationale, and we can take the next steps. Who's going to replicate? Could be the same investigators if you think of discovery and replication as being one step. It could be different investigators of the same school. It could be different investigators of competing theories or hypotheses. So some people more in the cognitive and psychological sciences have argued that you need contrarian replicators who don't believe in your hypotheses and therefore who do their best to kill it. Or combinations of the above, or you could make it open to the wide public. You could have open science, including even data and algorithms in your computers, and people could try to replicate what is being done. I'm not saying that this is the best solution, because obviously you need to have some qualifying credentials before you start going wild in trying to perform experiments or replicate experiments and results. Registration. Registration is another option, which means that we register ideas, much like we could register protocols, we register data sets, we register raw data, analysis, or whatever. There's different levels of registration. Level 0 research is unregistered research. I wake up at 3 o'clock. After midnight, I have some idea that I cannot sleep unless I go to the lab or I go to my computer and I start working on that. I'm sure that that happens to you all the time. It happens to me at least. I'm a little crazy on ideas. But, should I send a message to the entire scientific community that I have this idea, can you do that first and let me know the results? No. Probably I will go there and try to run some analysis on my computer, but then I have to be honest that that was an exploration. And others should try to validate it and make sure that what I did was correct. Level 1 is registration of the data set. So you register a data set that may contain many variables, and then everybody knows what is your power to perform analysis. So I register a data set with 3,000 variables, and I tell people what these variables are. It's like registering a nuclear arsenal. So people would know that Nectarius has 50 nuclear warheads that maybe he hasn't analyzed yet, but he could start pressing the buttons and running these analyses or conducting these studies. Level 2 is registration of a protocol. A protocol obviously is more than a data set, but what is included in a protocol can be very debated in several scientific fields. So level 3 would be not just the concept, but also the exact analysis plan. Level 4 would be a registration of the analysis plan and the raw data. And level 5 would be open live streaming, which means that I make my computer publicly available. You can see what I'm doing. You can send me a message in real time. John, don't run that chi-square test. It's wrong, you cannot run it on these data, you need to run an exact test. Or, John, this series of experiments that you're proposing to do after lunch, it's completely ridiculous, we have done that already, don't waste your time, do something else. Thank you, I'll enjoy lunch longer. How much of that registration do we need in different scientific fields? It's an open question. Maybe in many fields, especially in highly exploratory research, it could all be level 0. But even then, we would know that these results and these analyses were produced out of level 0, and therefore others would need to replicate it. How about sharing? Does it happen? This is an empirical study that we published a couple of years ago, looking at the 50 most highly cited papers, most highly cited journals across science. Many of those are biomedical, but obviously there's physics, chemistry, and many other sciences here. In terms of whether they have policies in place for sharing data, for sharing protocols, for sharing materials, whether that may be a prerequisite to publication. If you see green color, this is good news. And you see lots of green color here. So most of these journals do have policies in their instructions to the authors about the need to make protocols, data, software readily available to other scientists. But if you take a look at the last column, which probably you cannot read, it's tiny font, you have to trust me that there's tons of zeros there, which is how many out of 10 papers from each of these journals that we carefully looked in detail actually adhere to policies. So the scientific community is sensitized, but when it comes to real sharing, it doesn't really happen so efficiently. Along with colleagues from NIH, I'm repeating that exercise across the scientific literature. Hopefully, I would like to see that the picture is getting better. Now, I'm a big proponent for data sharing, because I don't think that data belong to anyone in particular. I think that maybe in the 17th century, the data perhaps belonged to the single scientist who generated them, because usually these people were funding their work out of their own pocket, and they were very rich, most of them. And it was like their pastime, and much like you don't want to share the chandelier in your palace, maybe you can say, I'm not going to share my scientific data. But for 2014, I don't think that is really defendable. But then, who's going to do it? When and how? It's not easy. And this is a paper that I wrote with Peter Doshi and Steve Goodman for Trends in Pharmacological Sciences about a year ago. It's a very long list. I'm not going to go through that, but it's 22 reasons why it's not easy to share data for clinical trials. Now, clinical trials is like at the end of the research and development story. It should be very easy to share data. It should be even publicly enforced that everybody, physicians, scientists, patients, should know why they're using treatments or not using treatments. Yet, it's not easy. And whoever wants could take a look at that paper on what kind of obstacles we need to go through. Let's say that we do have access to all the data in the world. And that's my dream. Everything that any scientist generates, any other scientist who's qualified could take a look. How would that world operate? So a few years ago, when I was still in IOANNIDIS, I got in touch with Miles Axland, the editor of Nature Genetics. I'm not even sure whether I did or he did. And Nature Genetics had started for many years a policy where all microarray studies, to be published, they had to have full data sets, full protocols, full software codes, everything publicly available as a prerequisite to publication. So we said, let's see if we can repeat the analysis that were published in papers with microarrays in Nature Genetics. We got together about 20 leading microarray analysts around the world. We created 4 teams of them, and we said, go ahead, try to repeat the analysis that led to that figure in that paper or that table in that other paper. In some cases, the analysts spent more than a month to try to repeat the analysis for a single table or a single figure. It was not easy. At the end of the day, they could repeat only 2 of the 18 analysis in this highest impact factor journal for papers by the very best scientists in the world. And in some cases they could reproduce bits and pieces, but with discrepancies. Most of the time they could not reproduce anything. And the reasons for that was that the data were not available, the software was homemade and it had disappeared, the methods were unclear, so some of the best methodologists in the world could not understand what's going on here, or everything was very clear, but the result was different. So this is what's happening under probably close to ideal circumstances. A couple of months ago, we switched to the other end of the spectrum of research translation, and we went to randomized clinical trials. So along with Shamil Ebrahim from my team, we said, can we find reanalysis of randomized clinical trials looking at the very same hypotheses and the very same analysis as the original publication. Now, these are trials that look at whether a treatment works or not, and the conclusion is that treatment should be used or should not be used, these patients should be treated or no. And we looked at whether the new reanalysis that had used the raw data reached a different conclusion. That no, you should not treat patients while you thought that you should, or vice versa, or you should treat different patients compared to what you thought based on the original paper. We found 37 such cases, which are very few compared to the hundreds of thousands of clinical trials that have been done, because data are not shared by default. And 35% of the time, the conclusion was different. So one might say that maybe these were reanalyses that were done by enemies or opponents or people who wanted to destroy the original study. Most of the time, almost always, the reanalyses were done by the very same authors who had published the first paper. Why did they reach a different conclusion? I think it's mostly the fact that the reward system is such that in order to have a paper published, they have to say that we found something new, something different, something that we had not seen the first time. So these people, I think, were even forced to try to generate a different conclusion just to get another paper published. Unless we can change that structure, it's very difficult to get a clear picture of whether we should just abandon clinical research if you take that seriously, or maybe abandon all research, which of course I do not believe in, or how are we going to fix these problems? Here's another empirical evaluation that I did with colleagues from IOANNIDIS and also from Edinburgh when I was at Stanford. Fotsis, who's still in IOANNIDIS, led that effort, looking at a very large number of thousands of studies of animal experiments and trying to see how many of those have some basic signals of evidence that suggest that everything is okay and I can take them to the next step, which would be human experimentation. These are animal studies of neurological diseases, and along with colleagues, we have tried to put together a collaboration where all scientists contribute databases and others that are not offered by the scientists or found through the literature on experimental studies on animal models. And as you very well know, there's hundreds of models of animals that seem to suggest that specific interventions work. When it comes to clinical research, you take things like stroke. Even though we have hundreds of promising interventions, we only have maybe one that has worked. I don't think we have a second. So many people say, well, animal research doesn't work, we should abandon that. I think that's the wrong message. Animal research could be highly relevant. But the problem is that the way that that evidence is collected has a lot of problems. So this is a difficult Venn diagram, but we tested the evidence for the presence of what we call small study effects, which is just bias, mostly publication bias, excess significance, which is also characteristic of reporting biases, small sample size, which again would give you a very noisy type of situation. And obviously, you need to have a significant signal. At the end of the day, only 8 pieces of evidence out of 140, which included several thousands of animal experiments, had no signals of biases or problems. So I think the animal models were relevant, but the way that the research had been done and communicated left plenty of room for improvement. Another issue is how to contain the influence of conflicted stakeholders and authors. There's many efforts that try to inform other scientists about potential conflicts of interest, and for some types of research, like randomized trials, meta-analysis, cost-effectiveness analysis, guidelines, and other sensitive studies that may affect translation into clinical practice, I would argue that everybody needs to know very carefully who has funded that research, who is doing what, and why, and is there some potential conflict of interest in that design or in that result or in that interpretation. This is a study looking at the most prolific pharmaceutical companies, and each one of them is a node. The size of the node is proportional to the number of trials that they perform, that they fund, and they sponsor. The connections, their thickness is proportional to the number of trials that have been co-sponsored by 2 or more companies. So if you see a connection between Merck and Amgen, it means that there's none here, or Merck and Abbott, it means that they have co-sponsored some trials that look at products that both of them have. And the autoloops, the thickness of the autoloops is proportional to the number of trials that these companies fund that pertain only to their products. So the autoloops here are far more thick compared to the connections. What that means is that for clinical trials, the vast majority of research is done specifically with the outlook of single products by a single company. They're more like marketing tools rather than scientific experiments. Are the companies to blame for that? I don't necessarily think so. I think they're forced by regulators to do this. They have to fund these trials. I would argue that it would have been better if a public entity could use these funds to design trials that would compare agents by different companies and treatments by different companies. Better statistics and methods. So there's many options, like transparent, registered statistical analysis plans, statistical training and improved literacy and numeracy of scientific workforce, better study design, standard features, for example, randomization and blinding of investigators in animal experiments, and maybe also some checklists that people could follow. NIH is experimenting with a number of checklists currently. Here's from a paper in The Lancet earlier this year where we had an assessment of trends in methodological quality indicators for in vivo studies. Most of those are animal experiments, and you see that for very simple things like blinding in the assessment of the outcome by investigators and randomization are still, even very recently, performed only about 20-25% of the time. These are very easy to do, the cost doesn't go up, it safeguards the validity of the results, but again, they're very infrequent. And when it comes to checklists, one has to think, how many biases do I need to think of and try to take care of? There's just too many biases that one has to think of. This is a paradigmatic proximity map that we generated with David Cheval-Rias, looking at 17 million PubMed papers focusing on biases. We identified 235 biases across the literature being discussed. I'm not sure whether I'm an expert on bias, but I have to say about half of them I had never heard of. And it's very important whenever a study is designed to try to think how many of all of these biases may be important, and therefore I need to be careful to optimize my design or whatever I do to try to bypass them. This is a time array for biases, not a microarray or equivalent. We have the 40 most common bias terms in the scientific literature. And there's obviously an error here, like in any scientific work. I have the right to perform to have many errors in my slides. 1882 is wrong, obviously. But
Confounding, for example, if you see black, it means that nobody is talking about it. If you see white, it means that lots of papers deal with confounding. But dealing with confounding does not mean that they eliminate it. They may just mention that confounding is a problem, but my problem doesn't have any problem with confounding. My study doesn't have a problem. Or same thing with publication bias. There's lots of use of that term, but it doesn't mean that they take care of publication bias. And they say meta-analysis could suffer from publication bias, but my meta-analysis is immune. Another possibility is to learn to live with small and tiny effects. So this is an increasingly common situation in medicine and the biomedical sciences. We're talking about odds ratios of 1.05 or less. Many of those would be highly relevant clinically for public health, but we have to run very large studies to be able to document them. One other option is to even forget about the possibility that there are large effects out there. In one empirical evaluation a couple of years ago that we published in JAMA, we looked at 220,000 clinical trials trying to see how common it is to find big treatment effects. When it comes to mortality, with highly statistically significant results, with replication in at least 2 studies, with no clear evidence of bias, we only found one intervention within 220,000 randomized trials and 85,000 meta-analyses that fulfilled these criteria. Extracorporeal oxygenation for premature babies. Obviously there's more. I mean, there's things that have not been tested in randomized trials because if I fall dead on the floor, is there any physician in the audience? I'm sure that there's a few. You will run to save me, even though my lecture was horrible. But this has not been tested in randomized trials, and it would be the best thing to do. But basically, we have to reorient our way of performing research towards capturing these modest but still very important effects that need very careful large studies to detect them. This is just a big picture from that analysis. Whenever we did document a big effect in the first study, whenever we did a second study, it was much smaller, or much of the time there was absolutely nothing there. When it comes to analysis, what we need to do is try to capture the extent of vibration of effects that exist in a given field. By vibration of effect, I mean how extremely different results one could get by changing the analysis plan. These are clouds of results. Each cloud has about 1 million points, and these are very simple analysis. It's one risk factor, or putative risk factor, in terms of its relationship to mortality risk. And these are the hazard ratios, and this is the minus log 10 p-value. So each association has been analyzed in 1 million different ways. How do we do this? Well, there's many other things that could affect mortality. So for each one of them, like age and gender and exercise and what you eat and what you do and whether you have any disease, you can include it or not include it in the model. So 2 times 2 times 2 times 2, if you do that 17 times for 70 factors, you end up having about 1 million different analyses for the same association. And much of the time you get that picture, like for vitamin E and mortality, you get some results that suggest that vitamin E is good for your health, and some results that suggest that vitamin E is bad for your health. And if you have a strong reason to prefer one result or another, you can just report whatever you believe is true, but this doesn't necessarily mean that it is true. I think I have run out of time, so I will try to close with a few more slides, and hopefully there will be some time for discussion. Improving research reporting standards, I think, is another interesting possibility. There's the Equator Initiative that has accumulated about 200 standards for reporting different types of studied results. There's many efforts in different scientific fields that are probing the reproducibility of specific research fields. The Center for Open Science was launched by Brian Nosek from the University of Virginia. They're testing large-scale psychological experiments that are influential in the literature to see whether they can repeat them and replicate them. The Reproducibility Initiative that was launched by Science Exchange is using independent labs currently to try to reproduce the most influential experiments and papers in cell biology and so forth. Our new center that I launched a few months ago with Sue Goodman the Meta Research Innovation Center at Stanford, we're trying to be a coordinating hub for efforts to improve reproducibility. To change the world, I think we may need to think about changing the reward system. Why do we do research? We do research because we enjoy it, because we love it, because research and science is the best thing that can happen to human beings. That's what I believe. But at the same time, there's funding, there's people who need to have a career, they need to put some food on the table. So can we reward the best practices in order to get the best practices? This is something that I argued with Moin Korea a number of years ago in science. And since then, there has been a lot of discussion. A few months ago, we published in JAMA a proposal to re-engineer the reward system. We call it the PQRST. It's like the electric car. To have a nice PQRST. So P stands for productivity. And many systems are looking at productivity, more papers. We argue, forget about more papers. We don't need more papers. We have billions of results. We need good papers. So focus on the top two, quality, and each field has to agree on what are the quality parameters. R, reproducibility, reward reproducibility. S, sharing, reward sharing of data, of protocols, of materials, of software, whatever one does. Give credit for that. And T, for people who are in applied research, reward translational impact. So it did make a difference. It did deliver something. People's lives were improved or saved, or that airplane did fly, didn't crash. To do this, one has to examine what would be the value of different items in the academic and research community. So this is from the PLOS Medicine paper, and the first column is the current system, where publishing is what matters. So you publish a good paper, this is 1 point. It doesn't matter if it gets refuted, if it gets translated, if it gets reproduced, who cares? Sharing, no value. Contribution to good quality peer review, no value. Contribution to good educational training, no value. You get a grant, you get 5 points. So you can trade 5 papers for 1 grant. An assistant professor is 3 points, an associate professor is 10 points, a tenured professor is 20 points. If you get a doctoral student, this is 2 points, so you can trade a student for 2.5 papers, or, did I say that correctly? 2 points for a student. No, an R01 grant is 2.5 students. Or 2 good papers you trade with 1 student. It's like a slave market. No, I'm kidding. Power, like the prime minister or the minister or the peripheriarchist, or I don't know what, that could be 200 points, up to 200 points. So now you can think of ways to change that. So you can win 2 points if you publish a good paper that gets replicated, and maybe 5 points if it also gets translated, but maybe lose 1 point if it gets refuted. You win if you share, if you contribute, teamwork, if you are a good educator of high-quality science, and maybe if you know the peripheriarchist or the prime minister has no value. And this is even more disruptive if you know the prime minister and the peripheriarchist, you lose 200 points. If maybe then even being an assistant professor versus a tenured professor or just a student has no difference, and we're all part of the same scientific community. Now, if you do that, maybe, and if you become more disruptive, you need to think about what would be the consequences and what would be the cutout or the damages. So I'm not saying that it's necessarily the best solution. And finally, one needs to understand and align the interest of other stakeholders. We're not alone in science. Scientists, I think, would believe that science belongs to us, but there's other people who are involved, like the industry, private investors, public investors, some of them open, others like the military that closed, journal editors, profit publishers, professional scientific societies, universities, not-for-profit institutions, supporting staff, administrators, hospitals, insurance companies, governments, federal, states, whatever authorities, and people, common people. What all these institutions and organizations and people expect from science is not the same thing. So some people want publishable results, others want fundable results, some want translatable results that work, and some just want to make profit out of that. So we need to understand who's out there, who's doing what, and try to align these interests to try to get to the truth and to something that would help other people. To conclude, there's many possible interventions that may improve the efficiency of research practices and the reproducibility of the scientific literature. Few of them have strong empirical support. Empirical research on research would be useful not only to assess the prevalence of problems, but also understand whether there's any harms in changing some of the systems that we think might be malfunctioning. And finally, the adoption of optimal research practices can be key to the success rate, efficiency, credibility, and legacy of scientific fields. As I said, science is the best thing that has happened to human beings, and it deserves that we should get the better out of it. I want to thank a number of my colleagues who had the bad fortune of collaborating with me. I'm a very bad collaborator, but about 3,000 people have suffered doing that with me. And I can only list a few of them who really need to get the credit for whatever empirical I showed you today. I would not have happened without them. Thank you.
Question.
Me? Okay, there are several issues one would like to discuss, because I think we are all described in order to generate some thoughts and discussions. First of all, irreproducibility in some sense is not really bad. For example, I know some experiments with microarrays produced by Beer in Amsterdam. I have tried to reproduce, it was impossible. Nevertheless, they built a company, Agentia. They are now dominating the market. And in order to do this kind of analysis, you have to ship the sample to them, and only them know how to provide the result. So, in that sense, the reproducibility builds a monopoly builds a company, makes profits. This makes the world go round. Okay? Another thing is that in order to try and avoid this kind of irreproducibility problems, we have to, as was suggested in many cases, we have to impose standards. But in order to do that, we need good scientists who know about what they're doing, and they are willing to spend time to generate standards, and then to suggest them for the community to follow them. But this is not disruptive science. So if you are as IT scientist, you would rather do data mining or than ontology, because ontologies are considered boring. And there are things like that I see that we are running after technological development, and technologies do not obey this kind of reproducibility laws. I mean, microarrays technologies, expression profiling started with SAGE. Now there are outcome transcriptome analysis. There is no way we can keep pace with these developments, and there is no way we can integrate this kind of different platforms. So I don't know. I mean, should we accept as this is being our fate? I mean, we have to accept that, yes, okay, there are going to be reproducible studies, and let's keep trying to find things.
These are all excellent thoughts, and I don't have like a very clear answer about that, because obviously the best answer may differ from one field to another. In general, I'm a little bit worried when we say that we can tolerate irreproducibility if a company or another institution can guarantee that they're doing it, and don't worry, I'll do it for you. I'll send you the samples and it will be done. There has to be some way that an outsider could cross-check that. Now, I wouldn't say that it's necessary to do it for every single piece of published work, because obviously that will become extremely difficult. But if that company is promising to use that information to treat patients or guide their management or really do something that could have an impact in real life and not just scientific curiosity, I would argue that we should have a mechanism that the technology could be checked for its validity and reproducibility, and also at the same time protect the fact that they should get the credit and therefore also the money for building it. So it's a very subtle point how to promote entrepreneurship and to protect the rights of the discoverer, but at the same time safeguard the validity of the science and also the utility for the application when it's applied science that is affecting human beings or our life. Should we try to replicate everything that exists in the literature? That would be a waste. I think that there's thousands and millions of papers that are just dead, and I would just let them sleep. But the problem is that some fields know exactly who's dead and who's alive. There's many fields that even if you ask the experts, they don't really know what's dead and what's alive. And if you ask two different experts, they would have very different views. They would say, no, this is perfectly fine. No, I disagree. So each field has to agree on what is the threshold and what is the threshold that reproducibility check should be engaged. And in some fields, it may be just 1% of all these results that may need to go through a very rigorous check. For example, I would say if something is heading towards the direction of application in clinical practice, in public health, in everyday life, I would say I want to make sure that I will not be mistreating patients with technologies that are harmful. So I want to make sure that it is really what they're promising. Or, I would argue, a highly cited research that is influential for guiding other paths that are taking that for granted and moving to the next step. I would want to make sure that this is correct, because otherwise you would have dozens and hundreds of thousands of scientists following a path of thinking that the previous step was as described, and that's not the case. So there's variability across fields on how to apply these rules, but I think that these would be my guiding principles.
Thank you for a very wonderful talk and stimulating. I would like to make a couple of points, thinking that a couple of them might be provocative, given that I'm in an institute of basic research. One of the problems, and you hit into that, is that there has been an emphasis, at least in the United States for many years, for the last 40 years, on basic science, basic research, molecular biology, and we're at the institute. If R01 does not have molecular biology, it's not going to be funded. And here it is, the big gap. Molecular biologists do it at the very, very bottom. And since I'm a psychiatrist, we are looking at a very complex behavioral phenomena. So because of the funding system and the pressure, they have to promise things that they cannot be promised. There's no good animal prototypes, models for mental illness. And again, our basic scientists, they promise for that. I'm sure you're aware, and many of you are aware of the so-called obesity epidemic in the United States. And this is in the last 30 years. And a lot of money, they go to genetic studies, to gene studies, molecular biology studies. But it's my opinion, it's very clear that I was talking with some prominent researchers in that area that psychosocial factors are much more important. Nothing happened in terms of gene mutation in the last 30 years to explain the obesity epidemic in the United States. So there is this overemphasis in basic research that is important, but it will take its effect, its impact 30, 40, 50 years later. And we need good clinical research. And clinical research is not just randomized trials. But this is my second point. A lot of clinical randomized trials are supported by the industry. And when we have institutes like agencies like NIH funding, many times do not reproduce these findings. The mental field is famous for such failures. A lot of new drugs, when the studies were conducted by a public agency like NIH, they did not reproduce the results of those studies supported by the private industry. Myself, sometimes I have tried in my own field of sleep, when I try to publish negative results funded by NIH, but then contradicting those by the industry, I have met with fierce opposition. And the last point, because other people may want to talk, is what about the negative results? We all of us know a lot of negative results cannot be published for various reasons, because of editorial policies, lack of interest, lack of novelty, but also because they're contradicting studies that they were funded by the industry. And maybe a suggestion will be, that negative studies that is worth being published, we should have a committee, not from the same field, but because there is a natural antagonism, not from the same agency, funding agencies, that they will look at them, review them, and see whether they are worthwhile being published. Because I think there is a lot of information that will be very useful to the scientific field.
All three comments are excellent. So the first one, obviously, is very sensitive, because if there's a given research pie, the usual debate is how much of that should go to basic research and how much of that should go to clinical research and how much of that should go to translational research. I'm not an administrator to make that decision, but I believe that you can have excellent research at all of these levels. So what I'm interested in is excellent research. And it could be molecular biology, it could be clinical, it could be translational, it could be... I have to say that some of the best ideas that I have met in my professional life on medical topics, they have come from fields that are not even considered within the mainstream of biomedical science. And I'm working with people who are behavioral economists, and working with computational sociologists, with mathematical sociologists, with informatics people, with even astrophysicists. and there's really no limit, no boundary that this is the field that is good science, that clinical medicine is good science and molecular biology is not so good fun science, or vice versa. Any type of science could be excellent, and any type of science we should try to support if it is excellent. When it comes to basic discovery, I would argue that it's the wrong policy to ask from basic scientists, no matter what they do, to promise that they will deliver results and applications. This is not basic science. If they can do that, they perform applied science, and this is wonderful, as wonderful as basic science, but it's not basic science. So I think for basic science, we should focus on the work being interesting, creative, carefully done, rigorously done by people who are committed, that have a track record, and the teams that they build, and try to build other people with to obtain their own track record in excellence. I think asking for deliverables, like you need to find a solution for schizophrenia with your basic animal studies or cell studies or whatever modeling and your science, you're asking these people to lie. Because we know that schizophrenia has been out there for so long. I wish that we find solutions to treat it and handle it and understand it. We are making progress, but I have seen so many grants where everybody's promising that by the end of the day we will solve the problem of schizophrenia, that it's just impossible to believe that they were correct. Clinical trials. Conversely, I have argued that industry should not be involved in clinical trials. It is as if you're asking a painter to go to a painting exhibition and his painting is hanging among the others. You know, he's drugged. And he needs to judge that work and say which painting is the best. What is he going to do? I mean, the industry is forced to judge, to evaluate their own products. They will do their best to make sure that their painting will win the competition and their drug will show to be the best. And as I showed you in that slide, this means that there's a fragmentation of research. Each company is just running a research agenda focused on promoting their product rather than understanding what really works best among multiple options. So I have argued several times, and I've published that all these funds for clinical research should go from the industry to a common public fund, and public non-conflicted investigators should design the optimal research agenda to understand the relative merits of all options that we have at any given point, and try to find really which is the best drug or which is the best intervention, the best device, the best technology, whatever that is. Negative studies. Negative studies are ubiquitous. We have many lines of evidence in many different scientific fields, including several papers. Even a couple of weeks ago, there was a paper in Science describing the problem of negative studies in the social sciences, for example. Social sciences have gone like a rebirth of reproducibility in the last few years. I'm giving a plenary in a couple of days in the BITS Symposium at Berkeley, which is an institute focused on improving reproducibility in the social sciences. It's not just clinical medicine. The basic notion that negative results disappear has been extensively documented again and again, even under the best circumstances. Just because the reward system is such that people don't get credit for negative results, that there's conflicts against publishing negative results, especially when it comes to money and products and drugs, willingness to suppress negative results being twisted to become positive, lots of spin biases and selective reporting, selective outcome, selective analysis reporting. I think that many of those problems could be solved by re-engineering the reward system and who's doing what. What is the expectation? Who should do that research and why are they doing it? If they're doing it for marketing, then research becomes advertisement. You're not going to open your TV and see an advertisement. This dental paste is the worst that you can use. Would you see that? So research should be dissociated from dental paste.
How do you comment on the fact that high impact journals, they encourage more trendy or fancy papers rather than actual scientific working sometimes?
So I'm not sure that I would blame the most influential papers, the most influential journals for kind of distorting the literature. It's pretty natural that when you have so many millions of scientists and so many millions of papers, if you have multiple journals, and there is a perception that one journal is the best or the more rigorous and has a higher standing, or people believe that it is the best, there will be more competition to appear in its pages. So we have empirically studied in clinical research the results of clinical research on the same topic when it's published in the 3 highest impact factor journals, New England Journal of Medicine, Lancet, and JAMA, versus other journals that are not so high impact, but they're pretty much studying the same question. And we tried to see whether the results are more exaggerated. When it was large studies that were involved, the results were identical on average. When it was small studies, the results were grossly exaggerated when they appeared in the high impact factor journals. So what that means for me is that when I see a paper in the New England Journal of Medicine, or Lancet, or JAMA, and it's a small study, not really a great study, and it promises something extravagant, I tend to be very cautious. If it's a large study and it's well done, that's perfectly fine. Can you apply that to nature or science? I think you can consider equivalent terms. If it's something that is a very thorough effort, very well designed, very well conducted, I would say yes, I mean, this is the best of the best, and I can take that to the bank. But if it's something that's really very extravagant, comes out of nowhere, and you have some questions about how it was done, I would say that the risk of non-reproducibility is pretty high. This is a very generic rule, and obviously you have to operationalize it for different fields.
I'd like your opinion about three topics that perhaps increase the risk of reproducibility. The first one is the software bugs.
What I mean with that is that we use software more and more to analyze big data. And recently we were analyzing some software regarding compiling behavior. We could see that most of the highly cited software, they return awful compiler warnings. This is the first thing. The second thing is that since the data becomes bigger, we have to use huge pipelines, which means 20, 30 software. The error definitely accumulates there. So I'm not sure what we see in the end. And the third topic is that usually, for example, when analyzing microarrays or when we see the results of microarrays, usually you see in the discussion parts of the paper that we believe these results because they make sense. So they make sense post-hoc. I'm not sure that this is a correct statistical practice actually, and I believe that any result could make sense, especially in genomics. Yeah, that's all.
Excellent questions and comments at the same time. So interpreted as comments, I fully agree on all three of them. Software can have problems of transparency. A lot of research that we do has multiple steps, and the caveats of each one of them with different software, different interface would multiply. So you may escape the first 4 rounds, but something may go wrong at the 5th round. And clearly, you can say that anything makes sense. I can give you an example from an empirical paper that we had published from IOANNIDIS, where we looked at microarray studies for clinical outcomes that were selecting and validating signatures of multiple genes. Typically, the signature would include 50 or 100 or even more genes. And then you look at the discussion section, what the investigators say about why these results, that signature makes sense. Now, if you have selected 100 genes, how are you going to discuss that? Ideally, well, but that would take a lot of space. You should look at each one of these genes very systematically, all the evidence we know about it, what it does, what it doesn't do in different systems, in animals, cell cultures, and so forth, and present that evidence. That is not done. So what people did, they're cherry picking one or a few genes, and they're discussing, and 95% of the comments in the discussion are in favor of what was found. That gene also does that, which agrees with. Only 5% we really didn't expect to see that gene. It's contrary to what has been seen. So 95% is in favor, and much of the time, it's very clear-cut that there's cherry-picking in the discussion about what genes to discuss. Sometimes, even though there's 100 genes that have been selected in the signature, the ones that are discussed are not among the 100, but relevant genes that simply have something to say about them that fits to the story. So the signature has found 100 genes, and one of them is estrogen receptor 1, but the discussion forgets about all 100 genes and says, estrogen receptor 1 was one of the 100. By the way, estrogen receptor 2 has been found in another study to be consistent with what we found. So it's the Wild West, it's pre-lunch, discussions of papers are in such state that I have published, I think a number of years ago, that discussion section should be eliminated. I think that the only part of discussion sections that makes sense is the limitations part. That should be stated. then maybe you can have an editorialist, another scientist, maybe an opponent scientist, or someone who would be objective, right in the discussion of the paper. So these are clear challenges, and coming back to the software question, the only way to fix that is to improve transparency about both the availability of all that software, how it works, how it operates, better education of their operators, and transparency on what exactly happened. And there's many initiatives in many fields that, in some of those I'm involved in, where we're trying to ensure that you can track everything that was done in the experimental process. So along with David Donahoe from Stanford, David has tried to make sure that you can have every scientific protocol and paper condensed into a barcode. And if you take that barcode, you can extract everything that was done about that paper. And it works. I mean, he has some papers that he has condensed to a single barcode. And you can have the entire literature condensed into maybe the space of that table surface. That would be nice. So there's lots of ideas on how to improve that situation.
There is an issue. 98% of the microarray experiments, as you said, they were wrong. And we were able to validate 1.2% of something.
These were genetic association studies.
Yeah.
Yeah, a little different.
But 98%. I cannot understand any methodological or any software problems. It is the subjectivity of the researcher who really wants to get something through and distorts everything to be competitive. Competition is not to distort everything in order to get there. And how about an honest guy who really wants to do proper work? He remains, it's like trying to go to Athens by car and compete with somebody who breaks all the rules of all the reds in all the lights, and you go and stop every time.
Yeah.
You have no chance.
So that means that we need to find ways to reward people who do the best research.
There's no such way. It's like playing football and then going to the negative. You reward the missing goals. It's impossible.
Well,
you go and then you reward the Mitroglou when he misses the goal. It's impossible, But I don't believe it, I try to be realistic, but the work is done in another way, You have to be honest by yourself. Otherwise, nobody's going to impose it.
So it's not necessarily an issue of honesty. I don't want people, especially younger people, to leave the auditorium thinking that scientists are fraudulent people who just try to cheat and the mere spirit of scientific investigation has distortion embedded into it. I mean, a good scientist is looking at a picture and is seeing more compared to a not so good scientist. I mean, he sees ideas and concepts and connections and possibilities that others don't see. So that part is okay, but simply one has to be transparent that what I show is what I describe as level 0 research. I show that which has not been documented before, which is novel, which is interesting, which could lead to some new path or whatever, but it's just the first observation, and I'm not yet claiming that I have found the solution. It may be so, it may not be so. Could someone please help and try to validate it? If we respect that part, and we do not ask for basic scientists to save the world all the time immediately, because as I showed, for many of the big discoveries, it took 20, 30, 40, 200 years to save the world. and much of the time the world was never saved. We cannot judge people by just putting pressure on them. You need to save the world in that grant within 2 years. It's not going to work.
They offer themselves to save the world. You know, I think everybody, these people, you know, they have the tendency to dominate and want to be prominent and dominant. Nobody asks anybody to do anything more than what one can expect from an average person. I don't know, maybe I'm wrong.
No. It's pretty much at the nucleus of what is causing these problems. Yeah.
Sorry? You have a question? Anybody else?
I think one practical way to be reproducible enough for some people collaborative enough, to collaborate with people outside of your institution and try to share your data and your thoughts and your hypotheses with other people and not be, you know, a solo guy. What do you think?
I think in many fields this has worked. So collaboration, team science, sharing of information in wide scale. In some scientific fields, this is really the only paradigm that works. If you take high energy physics, where the Higgs boson was discovered by a coalition of 40,000 people working together. Now imagine taking that paradigm, which did work, and the Higgs boson was discovered, and trying to take these 40,000 people and use a biomedical paradigm of the principal investigator siloed, sending in applications. So I'm one of them. I sent in my application. Aim to discover Higgs boson. Theo is sending his application. Aim to discover the Higgs boson. Yota is sending her application. My aim is to discover the Higgs boson. Within 3 years, I have to deliver, because otherwise I'm not going to fund it within 3 years. What would you get? My guess, you would get 40,000 discoveries of Higgs bosons, none of which would be correct. And we know that you know, each one of us would be collecting little bits and pieces of data right and left, and we would be publishing, I see a signal here, and here it comes, and none of them would be true, because now we know that unless all these people had coalesced their effort, it would not have been discovered. It was so difficult. So I think that is a good paradigm. We need to find ways to reward it, to make sure that credit is allocated according to real contribution, because then you can have collateral damages, you can have science becoming kind of a political enterprise, and some people just putting their name in papers with 800 authors doing nothing, and, for whatever reason. It becomes a new set of rules that we need to make sure that there's still transparency and allocation of credit and its own reproducibility check somehow.
So, based on the arguments that we've laid out, it seems that we have a fairly good idea about what actually to do in order to improve reproducibility in research. What would be your assessment, if you were to say? Are we actually improving? Are we reaching this idealistic state of really reproducible, reliable research? Or are we moving away from it and this industry is becoming completely uncontrollable because of its sheer growth?
It depends on the statistical measure that one would use to express whether we're improving or getting things worse. So if I were to guess, I would think that science is making more discoveries compared to what we had in the past. We have more knowledge, and the pace of accumulation of knowledge is increasing at some very good pace. If you look at how much noise and false results we have, I would say that also increases. And maybe in many fields it increases at an even faster pace. So maybe the proportion is even smaller of the good and valid and useful discoveries versus the 10 years of spurious or noise. So it is an evolving challenge, and it's not something that we can say, oh, we're losing the war or we're doing wonderful. And it's highly field dependent. There's some fields that are clearly improving, other fields that are very steady, and other fields that haven't heard anything about all of that discussion. I think that they're just continuing to have their professional societies and meetings and presidents and whatever, but you know, they're just becoming big pots of noise. So I cannot really predict that that field in 5 years from now will be perfect and that field will be worse. A field could dramatically improve like human genome epidemiology went from 1.2% reproducibility rate to 99% reproducibility rate because the rules changed. And we went from small single siloed studies with no replication to big genome-wide assessments with very careful, accurate measurements, teamwork, replication as a sine qua non. A completely new paradigm, 1% to 99% within a few years.
I have a last question, which is a little bit mean, but we were discussing here with my friend seniors. So the question is the research that you showed here reproducible? And because you publish it in a system that somehow suffers a little bit, and it reminds the fact that the saying that all scientists are liars. Big data, big noise.
Yes, and especially, this is the Epimenides paradox, or the Cretan liar paradox. Epimenides, famous Cretan of ancient Crete. But yeah, I'm sure that much of what I told you today is wrong. I have no doubt about that. And if it were not, probably I would be a preacher and, Christian, Orthodox, Muslim, I don't know, whatever, but I would not be a scientist. A scientist is one who makes errors. That's what defines us. We have the possibility of making errors. If we had no possibility of making errors, would be dogmatic politicians or something else. So, and I'm very happy to see that some of my work gets refuted or corrected or improved. Like in any other scientific field, empirical data and their validation go through the same process. So I'm more than welcome to see more studies and some of them overturning my early conclusions. And I obviously wish that some of these conclusions would be overturned with more optimistic estimates or better estimates. I don't think we should worry about making errors. This is really the strength of the scientific method, that it can identify errors and mistakes and correct them. So we should be perfectly happy with that. And I have even written a paper which is based on a commencement that I gave for Berkeley, that I'm very happy when I find that I have made errors, because the worst thing is the errors that I have not detected, but they're still there.
Related to that, for example, you published a quite intriguing study concerning the influential trees, and you have a formula that you have put the cut off in some. So is this formula, objective? I don't know. I mean, have you imposed some standards in order to follow that? I don't know. The question is now, if, let's say, we would ask a machine to do the same study with the kind of workflow that we generate unsupervised. What would be then the result? I mean, would the machine arrive at the same solution? Or should we promote this kind of approaches? I mean, having black box machines with workflows that they would do the kind unattended, and we would follow this kind of results, because we are taking part in the experiments, and we are influencing the results of the show.
So, that specific example is you can think of that as a diagnostic test. You run a diagnostic test whether someone has coronary artery disease or doesn't have coronary artery disease, you run a stress test, and it may be positive or negative, and maybe that person does or does not have CAD. No test is perfect. So when it comes to metrics of scientific performance, there's no test that's perfect. It could be that someone is found to belong to that elite group, and maybe others who are even better have not been included in that group because the test was not perfect. I'm pretty much in favor of quantitation. I don't know, I'm almost, I suffer from a quantitation disease. I feel very uneasy when I cannot quantitate something. So even when it comes to scientific appraisals, I believe that quantitative assessments do have a role. Nothing is perfect. They could be useful. They could become more sophisticated. Machines and intensive analytical modes could improve on that. I mean, there's like a variety of indices at the moment, and there's thoughts about how to combine them and how to like optimize. Nothing is perfect, but I believe that the direction would be to have more quantitation rather than have less quantitation. I'm very much interested to identify errors in different approaches, because there's some very clear missingness or errors here and there. So then you have to think, why did that go wrong? What was missed and why? And try to come up with better iterations. It's like in any scientific application, I believe.
Thank you very much.